Now, if this approach is applied to the cells, cell is a much more complicated system, so that's why it's very difficult to define the target molecule. But if you use the cell itself as the phenotypic screening, then still we can do something. So here, this is the stem cells. This is helping cells, so-called the feeder cells. Now, if your goal is just finding out stem cell selective probes, one possibility is before you ask what is the difference between these two cells, you can just use whole cells for the screening. So for example, one column you can just put non-stem cells. Another column you can put this is a stem cell only. Then by adding same amount of dyes, and then you can check whether stem cell is much stronger stained than the other cells. Maybe you can use this kind of very small plate I showed you. This is 384 plates. And then first column is a non-stem cell. Second column is a stem cell only. And third column is a co-culture. And then by adding same amount of dyes, and then you can take the high speed of the camera, you can take this kind of images. In these images, if only the first column is weak, and second and third one, which has the stem cells, if they are brighter, and these compounds has a chance to be stem cell selective probes. By using this kind of primary screening, you can select possible candidate molecules, and then by testing several other conditions, now you can make out this kind of probe. This is the first small molecule probe for embryonic stem cells. This kind of probes they call compound of designation 1. And every compound has a color, so this has a color code here, CD yellow 1. This is the first embryonic stem cell, the probe. Embryonic stem cell is similar to cancer cells. Once you isolate those cells, it's very easy to grow them. It's a very grow fast and also very robust cells. But time to time, if the embryonic stem cell is differentiated into the next step of a different tissue stem cell, so-called adult stem cell, for example, brains, the neural stem cells. Now, isolating is difficult, and also culture is also difficult. That's why if you test, now the target is a neural stem cell. And this is a stem cell, and this is a feeder cell. These are negative controls. Neural stem cell make several different kinds of brain cells. So astrocyte is one of them. So these three cells are negative controls. And only this cell is a positive control. Using same platform, using 384 well plate form, we can test very quickly and then find some compounds which stains only neuronal stem cells. By screening, we found this kind of the neuronal stem cell selective compound. Then we call this compound as a CD red 3. You can guess the color is red color, and this is the third compound they found. Here we have the stem cells, and stem cell differentiate into neuronal directions, but this neuronal stem cell is not pure enough. So that's why in this case, if you add CD red 3 here, then only neuronal stem cell is stained. Then you can isolate this neuronal stem cell and then utilize. In case sometime later, if your brain is damaged by accident or the disease. Now let's say stem cell differentiate into different tissue stem cell. One of them is a neuronal stem cell, and then neuronal stem cell make several different type of cells in the brain. If you take out those brains and then culture, there are many different kind of cells in there. But without the proper probes, it's very difficult to say which one is the neuronal cells or the different type of cells, and also which one is the dead cells. It's very, very difficult. If you have probes with the colors and specific to one type of cells, it may be very, very helpful. Now, in this case, we found two different kind of other probes. One is for macrophage, microglia in the brain, and the other one is for neurons. Microglia is very fast moving, so it can be very useful, but neuron is moving very slowly. That's why detecting or monitoring those neuronal cells or the motion has been very, very difficult. The only way so far has been this kind of GFP, green fluorescence protein method has been available. But to make this kind of a green fluorescence protein, you need to design and also engineer genetically. And then take those mouse and then mate them and then wait for the next generation of the babies. Kind of a long time of process is necessary. This looks like GFP stained neurons. Actually, this is small molecule probes added the case. In this case, by adding so-called neural orange are the probes, now you just culture those neuron cells. And by adding these other probes in the neural culture, now you can visualize directly this kind of a motion of neurons. This is possible. To see this picture more clearly, we reverted this color. So now it's the same movie, but in this case, this black color is a fluorescence signal. Now you can see this is one cell of neuron, and then neuron has multiple numbers of dendrite, and it has axons growing out to give out the partner. And this kind of real-time the imaging or the monitoring was not possible so far, but now it's possible by using this kind of small molecule, just to culture the cells. By adding this kind of a probe and then visualizing those motions is possible. For example, this axon is trying to find the partner, but eventually it couldn't find the partner. Sometimes it can come back to itself. Maybe it's a firing and exciting, firing, exciting. Kind of unnatural or maybe very strange phenomena is also to follow. Bye-bye.